All right, well, we are going to get started. Welcome and thank you for joining us for the ICR webinar for the month of April. I am Chad Kinney, the Director of the Institute of Cannabis Research, and I will be moderating today's webinar. Before we get started with the presentation, I would like to discuss a few housekeeping items. Today's presentation will be followed with a short question and answer period. To ask a question, please do so using the Q&A feature that you'll find at the bottom of the Zoom screen. You can enter questions at any time during the presentation. We will address as many questions as time allows up until 2 p.m. For any technical issues, please use the chat function. And like the question and answer function or Q&A function, the chat function can be found at the toolbar at the bottom of the Zoom screen. This webinar series is part of the ICR's initiative to broadly share cannabis-related research. The ICR also hosts an annual research conference. Beginning this year, the ICR is proud to be hosting the Cannabis Research Conference in partnership with our colleagues at the Global Hemp Innovation Center at Oregon State University. In fact, abstracts are currently being accepted for the conference, which will be held this upcoming August. If you're engaged in cannabis research and looking for a forum to disseminate your research results, the Cannabis Research Conference may be a good option for you. You can certainly find information about the conference on the ICR website, and we will post the link to the conference website in the chat window in a little bit. At this time, it's my pleasure to introduce Dr. Cristina Sempio, who will be delivering the webinar today. Dr. Sempio is a research instructor in the Department of Anesthesiology at the University of Colorado Anschutz Medical Campus. Dr. Sempio earned her PhD in forensic sciences at the University of Verona in Italy. Her doctoral research focused on the development of analytical methods for measuring drugs of abuse in a variety of biological matrices. Dr. Sempio completed an internship at the National Institutes of Health under the supervision of Dr. Marilyn Huestis prior to coming to the Anschutz campus as a postdoctoral fellow. Dr. Sempio supervises and manages the IC42 Clinical Research and Development's Clinical Toxicology Laboratory and is responsible for all industrial collaborations and analytics for cannabis and hemp-based drug developments. Dr. Sempio is collaborating on several projects to elucidate the therapeutic potential of cannabinoids and their influence on the endocannabinoid system. Dr. Sempio, thank you for joining us today. I'm sure that our audience, much like myself, is looking forward to your presentation. And with that, I will turn things over to you.
Thank you so much, Chad, for the kind introduction. Thank you, everybody, for joining, and a special thanks to the ICR for inviting me to give this seminar. As you can see, I'm going to talk to you about cannabinoids and endocannabinoids in human breast milk. I don't think I need to remind this audience how much the legal status of cannabis has been changed during the past few years, not only in the US, but worldwide. Still, the United Nations survey says that approximately 182 million worldwide use cannabis. Well, the most recent survey says that approximately 3 to 30% of pregnant and breastfeeding women are actually using cannabis at the same time. However, these numbers are rising because of the legalization, and this has been noticed both within state, comparing data from pre and post legalization, as well as cross-state, comparing data in the same year between states that legalized and haven't legalized yet. Data from the 2015-2017 National Survey of Drug Use and Health show that has been an increase in the prevalence of past 30 days use of cannabis among pregnant women. This increase does not only affect the overall number of women consuming cannabis, but even the number of times they use in the last 30 days, going from approximately once a week to almost twice a week. This increase could be even explained by a reduced perceived risk. So across 12 years, from 2005 to 2017, there has been an increase from 3.5 to almost 27% of women that are pregnant and believe that cannabis poses no risk for their pregnancy. And this data was not only among pregnant women that used cannabis before, but even from in between non-users. What is the which are the characteristics of these women? Well, most of them has a younger maternal age. They have a low income or socioeconomic disadvantages. They tend to have a co-use for tobacco, alcohol, and other drug of abuse, and are generally early in their pregnancy. A lot of the interviewed women says that once they realize they're pregnant, they tend to stop until the pregnancy is at delivery. The main reasons for using are nausea, pain, anxiety, and depression. And when we talk about pregnant women and nausea, one of the first things that comes to mind are morning sickness. So there has been two groups of researchers linked that decided to understand what was the response of dispensary at a woman inquiring on how to use cannabis to treat her morning sickness. The first study was performed in Colorado in 2018, and 90% of the dispensary suggest the use of cannabis, with 36% stating that cannabis is safe during pregnancy. Only 32% suggesting that the woman should talk with an healthcare professional without being prompted. The same kind of study was performed last year in Canada. 93% of the dispensary discouraged the woman the supposed woman on the phone to purchase cannabis to treat her morning sickness, and only 2% of them stated that cannabis was safe. 99% of the dispensary suggested talking to an healthcare provider without being prompted. Interestingly, almost all the dispensary interviewed in Canada have guidelines suggesting that pregnant women should be referred to healthcare professionals. But what are the possible effects of, what do we know for now, so until now, about delta-9 tetrahydrocannabinol, THC, and pregnancy? Well, we are all well aware that the amount that actually enter the bloodstream of a user depends on a variety of factors, such as the strain, the potency of the product, the kind of the product, the route of administration, and not last titration. However, there's a growing body of literature that show that during pregnancy, THC is able to cross the placental barrier and reach the fetal circulation. At the same time, there are data proving that THC can be detected in breast milk. However, the bioavailability for the infant is unknown. The possible effects of exposure to THC are mainly unknown, and these are not going to be discussed during this seminar because I really want to focus on what's happening in the breast milk. So what about breastfeeding? Well, the only survey I was able to find in the literature regarding breastfeeding professionals was performed on the participant at the 2014 Vermont Lactation Consultant Association conference. Out of 74 participants, 59 declared that they have been working with women breastfeeding while using cannabis. And overall, a median of each professional worked with a median of 100 women, out of which 15% declare cannabis use. The professional clearly were asked which statement best describes your current stance on women who breastfeed and cannot stop using cannabis. And as you can see, the answers are really different. The majority said that it depends on the situation of the woman they are interacting with. It depends on the severity, the frequency, the family situation. Some of them suggest that they should continue breastfeeding because the benefits of breastfeeding outweigh the harms of the cannabis use. And others said that they should stop breastfeeding for the exact opposite reason. So the majority of the association involved in breastfeeding have a fairly common ground, that is that breastfeeding should not happen if the mother keeps using cannabis due to the possible negative effect on the neurodevelopment of the child. However, if the mother is willing to abstain from cannabis use, then breastfeeding should be encouraged. So how THC can actually enter into breast milk? Human breast milk major components are protein, fats, and lactose, but it's a very characteristic sugar of the breast milk. However, the breast milk is an incredible biological matrix because it's able to adapt and change according to the prematurity state of the newborn, the infant age, the volume consumed, and the maternal diet. We can there are three major classifications for breast milk. The first one is colostrum, and colostrum is the first milk that the mother produces after delivery. As you can see, it's rich in protein, not that rich in fats, and it has a good amount of lactose. And over the following 2 weeks after delivery, the breast milk transition into mature milk. Mature milk is less rich in protein, has a higher concentration of fats, and it's more caloric and energetic than colostrum. If you look at the picture, you can clearly see that there's a difference between the 3 days milk that is colostrum to the mature milk at 25 days. A completely different matrix is the preterm milk. This is highly energetic, and it's the one that a preterm mother would provide for her kid. So there are several different ways that substances can enter into the breast milk. Exocytosis is the major route for the milk protein component, as well as for lactose and the aqueous fraction of milk. Lipid secretion, of course, is the one preferred by the fats that are present in milk, while apical transport and transcytosis are generally used from macronutrients as well as amino acids and other major proteins such as immunoglobulin. And how can THC take advantage of this system? Well, THC has a small molecular weight and is highly lipophilic. So the high fat content of the breast milk definitely attracts THC. And at the same time, the altered permeability of the mammary cell barrier allows a small molecular weight to pass across its barrier. So how much and for how long THC could be present in breast milk? The first answer to these questions was the one from Perez-Reyes and Wall in 1982. They were able to analyze a paired milk plasma sample from a chronic frequent user, and they established that the milk to plasma ratio was approximately 8:1. Thanks to questionnaire, they were able to estimate the time since last use as well as the approximate amount consumed by the mother, and they established that approximately 8% of the maternal dose per kilogram would be available in the breast milk and consumed by the newborn. They were even able to get samples of the infant feces, and they detected high concentration of THC as well as their major metabolites, 11-hydroxy and THC carboxy. This was a proof that the newborn was able to absorb, metabolite, and excrete THC absorbed through the milk. To get a confirmation about the possible milk plasma ratio, we had to wait this year. In January this year, Moss et al. Enrolled 20 chronic frequent users, and they asked them to provide a paired plasma and breast milk sample at 2 weeks and 2 months postpartum. They established that the milk plasma ratio was approximately 7, in line with what noted by Perez-Reyes. And they even noticed that the THC concentration increased over time, probably due to the differences between the fat concentration in colostrum and mature milk. Unfortunately, no data were available on the kids. In 2018, Bertrand et al. Were collecting samples for a breast milk repository in San Diego. They collected 54 samples and 34 were actually positive for THC. Through questionnaires, they were able to reconstruct time since last used and approximate amount consumed by the mothers. They were able to estimate that the average half-life of THC in breast milk would be 27 hours, with an estimated longest duration of 6 days. Unfortunately, no data from the kids were collected. Baker et al. In 2018 enrolled 8 women from Colorado that were asked to purchase and consume 0.1 gram of cannabis containing 23% THC. The women, they were then asked to pump their milk at 20 minutes, 1 hour, 2 hours, and 4 hours after consumption. The women were requested to inhale the product through smoking or vaping. This is very important because during a smoking event, the peak plasma concentration are reached within the event itself. However, when we look at the concentration profile in breast milk, we notice that the peak concentration are reached at 1 hour after dose, suggesting a time lag between consumption and accumulation in breast milk. The authors were able to estimate that the mean THC concentration of 2.5% of the maternal dose would reach the kid, and an estimate average absolute infant dose would be 8 micrograms per kilogram per day. Again, no plasma of mother nor kids were collected. In 2018, I was so lucky to be contacted by these 2 amazing researchers, Erika Weimar and Maya Bühnig. They were just awarded a Colorado Department of Public Health and Environment grant. Their aim was to perform an observational study that would involve women that at delivery would be positive for urine screening only for cannabis. The women would be enrolled at delivery and asked not to consume for the following 6 weeks. At the same time, they would provide paired plasma, urine, and breast milk samples 2 to 5 times per week. Unfortunately, due to the Colorado report law, they decided not to test the children. They were able to screen 400 women, and they were able to enroll 30, out of which 5 dropped out. Of these 25 enrolled women, however, only 12 were actually able to maintain abstinence throughout the 6 weeks of the studies. The demographic of our cohort was in line with what previously reported in the literature, with a younger maternal age, a lower education, and a marital status of single or a weird family situation. When these women were asked about their frequency of marijuana use and specifically about if they were consuming 2 times or more per week, categorizing them as a chronic users, most of our subjects actually could be considered chronic users. Their preferred method of inhalation was smoking or vaping. Our lab recently developed 2 analytical assays using liquid chromatography tandem mass spectrometry for the detection of cannabinoids in human plasma, urine, and breast milk. So we used the LC-MS-MS assay we validated for the analysis of the breast milk samples obtained from the clinical study. And here you can see a summary of our results. Interestingly, 187 samples were positive for THC, with a maximum concentration of 170 nanogram per ml. Only 24 samples were positive for CBD, with a maximum concentration of 14 nanogram per ml. Very interestingly, only 30 samples were positive for THC carboxy, and the concentration were very low, with a maximum reaching 3 nanogram per ml. To determine if our participants were abstainers or not, we decided to plot the milk THC concentration versus the plasma THC carboxy concentration. And as you can see, there's a clear correspondence between these graphs. And it's even clear which category our subject would fall into. This is an abstainer with a nice pattern of descending concentration, while our late fail abstainer had a clear spike in both THC milk concentration and THC carboxy plasma concentration at approximately 5 weeks. The continued user didn't change, as well as the failed abstainer. It actually increased in concentrations. we were still able to obtain a full set of data throughout 6 weeks for 7 women that abstained. Interestingly, a week postpartum, the median THC concentration were 3 nanograms per ml. This concentration increased 2 weeks postpartum to 5.5 nanograms per ml. And then most of the sample were still positive at 6 weeks postpartum with a median concentration of 2 nanograms per ml. On the abstainers, the milk to plasma partition was 6:1. However, the milk to plasma ratio might be greatly affected by other factors, such as time since last use, dose, and route of administration. So our 6:1 ratio is strictly related to women that abstain. Based on our data, we were able to estimate the half-life of THC in breast milk to be approximately 17 days. However, this means that the projected time to elimination is greater than the 6 weeks of our study. And here you can see the semi-logarithmic plot of the pharmacokinetic modeling to estimate the time to elimination. As you can see, especially in subject 7 and in subject 4, there are increasing concentration of THC over the first 2 weeks, followed by a prolonged period of decreasing concentration over several weeks. This could be due to a lot of different reasons. Sorry. Okay. I apologize, my computer froze. And PowerPoint crashed. I'm really sorry for this.
Perhaps might want to try restarting PowerPoint and see if you'll be able to open it again.
Yes.
In the meantime, I'll reiterate that if you have questions that you think of as the presentation is going on, please feel free to enter those into the Q&A function in Zoom. Again, that is located where you can open that function at the bottom of the Zoom screen.
Is it back up?
It's back up.
Okay, good. I apologize. So as I was saying, our pharmacokinetic modeling showed that concentration increased over the first 2 weeks and then decreased over a prolonged period of times. Okay, it does not like changing this slide apparently. Okay. This phenomenon was probably due to the changing fat composition between the colostrum and the mature milk. And at the same time, it could be related to individual metabolism, as well as differences in the marijuana use of the mothers before delivery. Limitation of our study were the inability to strictly quantify the amount of THC consumed by the mothers, as well as difficulties in abstention. Only half of our subjects were actually able to maintain abstention, and only 7 up to 6 weeks. And unfortunately, we had no infant data. So overall, we can prove that THC accumulates in breast milk during lactation. It is excreted in breast milk up to 6 weeks if the mother is a chronic frequent user but kept consuming throughout the pregnancy. Methods such as pumping and discarding only shortly after consumption might not be very effective. For this reason, several hospitals now suggest that in case of cannabis use throughout the pregnancy, pump and discard should be done for at least 2 weeks while the mother is abstinent before breastfeeding can start again. However, I think that it's absolutely clear that more clinical studies are needed to establish the safety of cannabis consumption during breastfeeding. I would like now to spend a few minutes talking about breast milk banks. In the past year, there has been a growing body of literature proving the importance of breast milk as the main nutrition for all newborn and especially for preterm infant. Unfortunately, only a third of preterm mothers are actually able to provide milk for their newborn. So associations like the Human Milk Bank Association of North America and the European Milk Banking Association are born with the mission to provide safe milk from donors for any newborn that might need it. This association had strict guidelines regarding the safe expression, handling, pasteurization, and storage of breast milk to guarantee that it will arrive safely from the donor to the newborn that actually need it. Unfortunately, there's a lack of regulation regarding how to screen for drug of abuse, as only questionnaires are requested. However, some breast milk banks decide to test their donor milk using ELISA kits. Our local breast milk bank, that is situated in Arvada, uses a Neogen THC forensic ELISA kit for analysis of THC. This kit is a one-step qualitative kit for the detection of THC carboxy. The kit has been developed for urinary screening of THC, and THC carboxy is the major urinary metabolite of THC, thus making this screen having 100% reactivity for THC and only a 4% cross-reactivity for THC. We decided to analyze a subset of 30 breast milk samples from our clinical study using both the ELISA kit and our LC-MS assay. When analyzed with the ELISA, all samples were reported as possible positive hits, with absorbance between the negative and the positive cutoff that was set at 5 nanogram per ml. However, quantification was not possible. When analyzed using LC-MS, all samples tested positive for THC, with concentration ranging from 0.9 to 130 nanogram per ml. Interestingly, only 7 out of the 30 samples were positive for THC carboxy, with concentration very low with a maximum of 3 nanogram per ml that is below the cutoff set by the ELISA kit. So we can say that the ELISA kits that are developed for urine analysis are not an effective tool to detect cannabinoids in breast milk because they focus their attention on the detection of THC carboxy as the major urine metabolite, while THC is the component that accumulates in breast milk. For this reason, a more sensitive method such as an LC-MS should be used to test breast milk samples, or a brand new kit should be developed from scratch. I would like now to focus a little bit on endocannabinoids. Their role in development has been proved more and more over the past few years. The endocannabinoid system seems to be supervising the proliferation, migration, differentiation, and survival of the neuronal cell during the neurodevelopment phase. However, the homeostasis of endocannabinoid has been proved important even for the health of the neurological system during adulthood. For instance, it has been proved that anandamide levels, that anandamide is one of the major endocannabinoids, is increased in Parkinson's disease subjects. And drugs such as levodopa and to a certain extent CBD are actually able to reduce the anandamide level, improving the symptoms of the Parkinson's disease. Another area in which endocannabinoids play an important role in the newborn are the appetite and the food intake. They are particularly important in establishing the suckling response and the satiety signal. In 2001, the Mechelen group decided to test the role of the CB1 receptor in food intake. Cannabinoid 1 receptor is the major cannabinoid THC receptor, and its activity is mediated by anandamide. However, the researcher used mouse pups, and they decided to inject them twice daily with a full antagonist of the CB1 receptor. That's name is SR141716A. In this case, they noticed immediately that after 6 days, all the pups were dead, and they did not gain any weight from birth, and they were not able to properly feed from the mother. So they wondered if THC would be able to at least partially reverse this situation. And they actually noticed that THC itself, its does not influence the overall survival or weight gain of the pups, and when it's given in combination with the antagonist, it actually helps reverting the situation with a better overall survival of the pups treated. So the investigator wondered if maybe injecting these pups twice for a week could have caused toxicity. So they decided to inject only the antagonist only once, either at day 1 or at day 2. And they noticed that the pups injected at day 1 were dead by day 4, while the one injected at day 2, 50% of them were able to survive, suggesting that the CB1 receptor plays a great role already in the beginning and at the establishing of the suckling response and of the food intake of the mouse pups. So they wanted to go deeper into this situation, and they wanted to see if co-administering the antagonist with the so-called entourage would improve and revert this situation. The entourage was composed of a mixture of 2-arachidonoyl glycerol, 2-AG, that is the second major endocannabinoid, 2-palmitoyl glycerol, and 2-linoyl glycerol. And they noticed that although the entourage was not able to fully revert the situation, it definitely improved the situation. The interesting observation was that it was difficult for the pups to actually suck and attach themselves to the mother, although the attitude of the mother was absolutely caring, and were actually spending more time with these pups than with the rest of the litter. Another important endocannabinoid involved in appetite is oleoylethanolamide. This endocannabinoid has been proved to act peripherally and be involved in the state of satiety, prolong the intermeal intervals, it reduced the size of feeding, increased the fatty acid uptake, delayed the gastric empty and intestinal transit. In 2018, Brun et al. Enrolled under 100 women from a cohort of women in the Netherlands, and they asked them to provide a breast milk sample at 4 months of age of a newborn. The women were chosen based on the weight for age Z score of their newborn. 50 of them had a high WAZ and 50 of them had a low WAZ. Interestingly, lower concentration of OEA, stearotyl ethanolamide, SEA, and palmitoyl ethanolamide, PEA, were actually linked to a high WAZ score and a higher weight gain since last birth. This might be particularly interesting because these two factors are linked to the possibility to become overweight or obese in adolescence as well as in adulthood. From the study, however, they observed that these three endocannabinoids might play a major role in the regulation of the appetite and food intake of the breastfed infants, and their level might have repercussions even throughout adulthood. In 2018, Gaitan et al. Enrolled 24 women that are non-users and asked them to provide a breast milk sample at 2 weeks and 4 weeks postpartum. They analyzed the samples for 3 fatty acid precursor and 12 endocannabinoids. I'd like to point your attention especially to the level of palmitoyl ethanolamide, PEA, and to the changes in the level of the 2-AG endocannabinoid that increased from the transitional milk to the mature milk. We recently developed a LC-MS assay for the analysis of endocannabinoids in plasma, and we decided to use it even for our breast milk sample. So our preliminary data showed that most of our endocannabinoids analyzed are in line with the reported data from the literature, except for PEA that has significantly higher concentration compared to the one recorded in the literature. I'd like to remind you, the previous study had an average of a median concentration of 0.9 nanogram per ml. We don't know, however, if this might be an artifact due to the sample storage. We did not analyze this sample right away for endocannabinoids, or maybe it could be an effect of THC. On the other end, we looked at the 2-AG concentration, and we noticed that 1 week postpartum were lower than 2 weeks postpartum and further increased during the following 3 weeks. So our observation was in line with the trend observed by Gaitan that 2-AG concentration increased over time and with the newborn development. However, we noticed that the maximum level that we reported were significantly higher, even up to 10 times higher than what has been reported by the other researcher. And again, we don't know if this could be an effect due to the sample storage or a presence of the THC in the breast milk. When we did statistical analysis on our sample, we even noticed that the concentration of linoyl ethanolamide, or LEA, actually have a trend and seems to be following a specific pattern when plotted against THC. And in this case, the pattern seems to be present both when the concentration are analyzed as they are and when the concentration are normalized to the baseline values. So we proved that endocannabinoids are an essential component for breast milk, and they seem to play a role in the food intake of the newborn. However, no literature is available to show if THC consumption can actually affect the level of endocannabinoids present in breast milk. Furthermore, and more importantly, there are no studies that clearly identified if an altered level of endocannabinoids might have an effect, or in which kind of effect, on the developing brain. So I think that the take-home message is definitely that more clinical studies are needed. I did a funny little research the other day on Google, and I just entered cannabinoids in breast milk. And these are some of the pictures that I got in the image section. It's mildly concerning that most of these pictures suggest that the same cannabinoids present in industrial hemp are present in breast milk, without distinguishing between endocannabinoids and phytocannabinoids. However, thankfully, I even found someone that reads our paper and understands and try to do a proper information of mothers that look online. I would really like to thank you all for your attendance. I would like to thank you, my mentor, Dr. Uwe Christians and Jos Klauter, as well as Erika Weimar and Maya Bunig. Thank you all.
All right, thank you very much, Dr. Sempio. I appreciate the information, and that was a wonderful presentation. We have had a few questions come in over the course of the presentation, and there's still time to submit questions through the Q&A function. The first question is, how does the bioavailability of THC in breast milk compare to other matrices?
So THC is generally, when consumed, THC immediately disappears from the plasma compartment. So that's why THC is generally used as a marker of very recent use, because it immediately, because of its lipophilicity, it tends to immediately go towards more compartments that are more rich in fat components, including the brain. So it is probable that breast milk, because of its high fat concentration, definitely attracts THC. And so that might explain the increased milk to plasma ratio and the fact that sometimes we were not able to detect THC in the plasma of the mothers while it was still present in the breast milk. That's why, to distinguish the abstainer from non-abstainer, we actually use THC carboxy as a marker, a longer marker of THC usage. I hope this answers the question.
Thank you. The second question is, in the longitudinal body mass study, how did you monitor for cannabis abstinence? Was there any other substance used by the mother? So I guess a two-part question.
Okay. So one of the requirements to be enrolled in the study, it was to be an only cannabis user. So the mothers were screened before enrollment to make sure that they were not using other concomitant drugs of abuse, especially. And we didn't look into the body, the BMI, but it's definitely an interesting point that should be addressed.
I think that was a mistake on my part. I used BM in the question, which I'm sure after hearing myself say it should have been breast milk.
Okay, can you repeat then that part of the question?
Yeah, in the longitudinal breast milk study, how did you monitor for cannabis abstinence?
We actually initially trusted the mothers, and then we just analyzed the sample. That's why the problem was that enrollment was really difficult, because we were asking the mothers to give so many sample in a week. And some of these mothers had issues with transportation and organizing and all these kind of things. So we prefer to be as inclusive as possible and keep as many mothers as we could, and just monitor the plasma and the plasma THC carboxy concentrations and the breast milk concentration of THC to prove if they were abstinent or not. We are actually thinking about publishing the data even for the non-abstainers, because we noticed very interesting things, such as that the milk plasma ratio could be even like 10 times higher than what we reported for the abstinent.
The next question is, does Dr. Sempio and her collaborators have plans to study the effects of breast milk THC on brain cognition?
There's another great researcher on campus. Her name is Tessa Krum, and we are collaborating with her, and she's doing functional NMR on the newborn to try to understand if there could be an effect. In her case, the mother she has a group of mothers that used through pregnancy and another group of mothers that were non-user as a control. And their object is to compare the functional NMR between these two groups to see if there's a objective measure that could help us understanding better this issue.
Great. A question about ELISA came in. Could sample preparation for the ELISA screening be adjusted to improve the accuracy for the breast milk matrix?
I think that the main problem is that the antibody coated on the ELISA kits are looking for THC carboxy. So the structure of the antibody is specifically looking for the chemical structure of THC carboxy, but is ever so slightly but different from the one of THC. And so probably maybe changing the preparation might help, but I don't think there's gonna be a big difference because of what the antibodies is looking for.
Okay. Next question. What effects do cannabinoids have on the endocannabinoid development functions?
More research are needed.
Okay.
I think that is, yeah, that's absolutely something that we have no idea about. And for me, it was very surprising that our 24 CBD positive samples are the only one that I could find in the literature. So CBD is available in Colorado at least. And at the same time, though, we have no idea about if it enters the breast milk, if it's absorbed by the newborn that breastfeed. So I think that this is really major field that needs to be studied. And there are some information, but I'm not an expert on it, so I don't want to. I think it might be a great topic for another conference seminar.
Okay. Have a couple more questions. Do the mandatory reporting laws that restricted you from ethically collecting infant data apply to all maternal drug consumption studies, e.g., alcohol, nicotine, or is this a marijuana specific issue?
It applies to federally illegal drugs. So alcohol and tobacco are not federally illegal, but cannabis is still, even if in Colorado is legal, it's federally illegal. And at the same time, so it applies to cocaine, methamphetamine, and all these kind of drugs. We now, the NIH, to try to help research, now has a very binding and very strict confidentiality process that could be started, and that the states recognize this NIH binding contract between the researcher, the PI, and the mother. And so maybe in the future, we might be able to get more data from the newborn as well.
Okay. The next question goes back to the start of your presentation when you were talking about responses by folks at dispensaries by the supposed pregnant woman. So are there key factors that you're aware of for the difference in the responses by dispensaries to inquiries about use of cannabis during pregnancy between the US and Canada?
I would first of all make clear that maybe the situation in Colorado changed since 2 years ago. That's something that I don't know. I know for sure that the Canadian government tried to make more strict rules. And I think that is reflected by the fact that in most of the dispensary, there was a guideline saying that pregnant women should be referred to a healthcare professional. So I think, as I said, I don't know if the situation changed in Colorado, but I think that the Canadian government tried to, based on the experience of the other state that legalized, try to place more strict boundaries.
Okay, and this is the final question that has come in. Are you aware of any studies suggesting changes in the birthing process in mothers who have recently used marijuana? For instance, related to anxiety to the fetus, ease of pain in the mother, are examples.
I am not aware.
Okay.
And so I cannot really, I'm sorry.
That's okay. All right. Well, thank you so much, Dr. Sempio. I appreciate your time, the research that you're doing. It's very interesting stuff. And as you pointed out, there's a lot of areas of continued research needs related to this topic. And hopefully you and your colleagues will continue to seek the answers that are desperately needed.
Yep. Thank you so much for inviting me, and thank everybody for attending.
All right. With that, we will conclude this webinar. Thank you so much for joining us.